we are going to know about the transmembrane system. What we are going to know? The transmembrane system. Uh, First of all, let me tell you why that system is necessary. By this transmembrane system, what the organelles are helping in the proteins, some essential materials through the Golgi body, endoplasmic reticulum, and the lysosomes. So these things will helping in the functions of the lysosome. What are the organelles are involved in these transmembrane systems? We need to know. First organelles is Golgi body, or you can say Golgi apparatus, and second one will be ER, that is endoplasmic reticulum, obviously rough endoplasmic reticulum, and third will be lysosome. Some of you may correlate that this transmembrane system is actually like as GERL system. It is not actually GERL system. It is actually only transmembrane system. The transmembrane system is helping in the GERL system. Without transmembrane system, GERL system will not be work. This about this transmembrane system, that this one actually beyond the NCRT. So these things is required for the NEET aspirant students, those who are going to appear for medical entrance. Now see, first of all you need to know about the nucleus. Let me give you some background of these systems. And that nucleus, we know nucleus is bimembranous and it is having some nuclear pore, and from the nuclear membrane that endoplasmic reticulum will form. That endoplasmic reticulums, actually I am not going to clear, that endoplasmic reticulums will be having some vesicles. This is actually called as cisternae. From here some tubules will arise. That tubules actually not embedded with the ribosomes, but here some ribosomes you have seen. This is about endoplasmic reticulum, and now here there will be Golgi body. That Golgi body will be having vesicles and vacuoles, and here are two phases. This is cis phase. This phase is for cisternae phase, and this is actually called as trans phase, which will be transforming phase of the vesicles. And this is also called as convex site, and this one is called as actually concave site, and this one also called as what? Cis phase, and this one is trans phase. So why I am going to draw? Because without knowing this, you cannot understand about the transmembrane system. And let me explain about the transmembrane system. Now see, this is suppose rough endoplasmic reticulum, and this is suppose this one, and here will be the nucleus. The positions of the nucleus is suppose here. This is the positions of the nucleus. Now in these mechanisms, what are the protein vesicles will be involved? COP2 vesicles, COP1 vesicles. This is COP2 vesicles, this one is COP1 vesicles, and this one is clathrine vesicles. Now question is, what is COP2, COP1 and clathrine? These are actually protein code. If you simplify it, COP is what? COP is coat of protein. COP is what? Coat of protein. And clathrine is what? Clathrine is one kind of protein which will form the cover of the vesicles. Now what happened in this phenomena? See, here two mechanisms will be taking place. One is anterograde transport, another one is retrograde transport. Now question is, what is anterograde transport and retrograde transport? Suppose we know what nucleus is, brain of the cell. So nucleus is actually giving signal to the endoplasmic reticulum to do some things, modifications in case of the inside the vesicles. So that nuclear or raw materials will be passing through the endoplasmic reticulum, and then it will be transferred into the COP2 vesicles. That COP2 vesicles, by the process of anterograde transport, entering into the cis Golgi network, which is actually cis phase of the Golgi body. This, from this cis Golgi network, it will enter into the median Golgi or midline Golgi, and then later on it will entering into the trans Golgi. Here transformations of the vesicles will be taken place, and if it is necessary, then it will be remain in the cell, or if it is unnecessary, through the exocytosis it will be released. This is the function of COP2 vesicles. Now during these procedures, if that nucleus thought that proteins is necessary, then after modifications in the Golgi body, that proteins will be entering into the another vesicle. That vesicle cover will be formed by another protein which is called as COP1 protein, so that it will form one vesicles which is called as COP1 vesicles, and again it will transferring into the endoplasmic reticulum, which is by the process which is called as retrograde process, isn't it? Or you can use retrograde transport. So this is about the COP2 and COP1 vesicles. Now another thing is, what about Clathrin vesicles? So Clathrin vesicles, actually the vesicles which is formed by the protein. The protein name is Clathrin protein. Now suppose cell need food. Cell needs what? Cell needs food. If that, we know what about the endocytosis? Endocytosis and exocytosis. So cytosis. So during taking the food, cell will do the endocytosis procedures. Through the endocytosis procedures, it will enter into the cell, and then it will form one vesicle by the helping of the protein which is called as Clathrin protein, and will form Clathrin vesicles. That Clathrin vesicles will be entering into the trans-Golgi network, and then depending on it, it will be going through the retrograde and anterograde transport mechanisms. And also these Clathrin vesicles can form the lysosomes. That lysosomes, we know there are different types of lysosomes: autolysosomes, primary lysosomes, secondary lysosomes. On the basis of the functions and on the basis of the situations, that lysosomes will do the work. This is all about the transmembrane systems. So that transmembrane systems, it will not follow properly, then GRL system also will not work properly.